Good morning, dear children. Today we are going to study Gynoecium or Pistil. Gynoecium, it is the female reproductive structure of the flower. It is formed of carpels. The number of carpels may vary. When there is only one carpel, it is called monocarpellary. When two carpels, bicarpellary. When three carpels, tricarpellary. When three or more carpels are there, it is known by the name polycarpellary. And the carpels may be united or free. When the carpels are free, it is called apocarpous condition. And when the carpels are united, as seen in the case of hibiscus, ladyfinger, etc., it is called syncarpous condition. A typical carpel has got three different parts, namely stigma, style and ovary. Stigma, style and ovary. Within the ovary, we can see the ovules are produced. And the ovules are the megasporangium within which the female gametes are produced. We have to study the structure of ovule. The young ovule is a small structure erased on the placentum. And the arrangement of ovule within the ovary along the placentum is known by the name placentation. And here the ovules are attached to the placentum by means of a stalk called funicle. This stalk, funicle. And the point of attachment of the funicle to the body of the ovule is called hilum. And each ovule has got two integuments, coverings. This is the outer covering and this is the inner covering. And these two integuments later on develop into the seed coat. And within the ovule, we can see there is a homogeneous mass of tissue called nucellus. And the basal part of the ovule is called chalasa. And towards the opposite side end, we can see within the integument there is a minute opening, and that opening is known by the name micropyle. And in the young ovule, we can see the central region is occupied by a homogeneous mass of tissue called the nucellus. Thus we can see the ovules are small structures present inside the ovary and attached to the placentum by means of a stalk called a funicle, and the point of attachment of the funicle to the body of the ovule is known by the name hilum, and the basal part of the ovule is called chalasa, and the two coverings of the ovules are called the integuments. The integuments later on develop into the seed coat, which encloses a homogeneous mass of tissue with inside is called nucellus. Nucellus, it is rich in reserve food materials. And when the ovule mature, we can see within the nucellus an embryo sac or female gametophyte is formed. Embryo sac or female gametophyte is formed. Later on we have to study the development of gametophyte. Here we can see within the mature ovule, embryo sac or female gametophyte is enclosed by the integuments. And we can see a portion of this nucellus remain as such in certain seeds. And that remnants of nucellus is known by the name perisperm. Perisperm. Megasporogenesis. Within the young ovule, the homogeneous tissue called a nucellus is there, and which is rich in reserve food materials. And one among the nucellar cell towards the micropylar end enlarges by absorbing nutrients from the surrounding cells, and that cells become the megaspore mother cell. And this megaspore mother cell thus formed from the nucellus is diploid in nature and it undergoes reduction or meiosis and form a linear tetrad, that is four cells are arranged in one row called linear tetrad of megaspores, and out of the four cells thus form three degenerates and only one become functional, which form the functional megaspore. And the formation of megaspore from the megaspore mother cell is known by the name megasporogenesis. The development of megaspore or functional megaspore from the megaspore mother cell by reduction division is known by the name megasporogenesis. And this, here we can see the megaspore mother cell, which is diploid in nature, undergoes reduction division and form a linear tetrad of megaspores, out of which we can see three cells degenerates and only one remain functional, which develop into the functional megaspore. The formation of megaspore from megaspore mother cell is called megasporogenesis. And this functional megaspore later on develop into the female gametophyte or embryo sac. Development of embryo sac or female gametophyte. And here you know the functional megaspore now undergoes three successive nuclear divisions resulting in an 8-nucleate embryo sac. First you know, the nucleus of the functional megaspore divides into two, forming a 2-nucleated embryo sac. Then the second nuclear division resulting in a 4-nucleated embryo sac. That is followed by the third nuclear division resulting in 8-nucleated embryo sac. Now we can see one nucleus from each pole migrate to the center, and these nuclei are called the polar nuclei. And the three nuclei at the chelasal end develop into antipodals, and the three nuclei at the micropylar end by wall formation develop into egg and synergids. These together known by name egg apparatus. Thus, a fully developed embryo sac is 7-celled and 8-nucleated. The 7 cells are by wall formation. We can see 3 antipodals by wall formation develop into 3 cells. Then the 2 polar nuclei within this large cell form 1 cell, and towards the micropylar end egg apparatus by wall formation forms 3 cells. Altogether we can see it is 7-celled and 8-nucleated. The 8 nuclei are antipodal nuclei, polar nuclei, and the 3 nuclei of the egg apparatus. Thus 8-nucleated. Thus a fully developed and fully mature embryo sac, we can see the polar nuclei are also fused together, forming the secondary fusion nucleus. Thus a fully developed embryo sac is this one. Then you know, within this embryo sac, we can see within the egg apparatus, the 2 helper cells called synergids are there. They are having some certain cellular thickening towards the micropylar end, which is known by the name filiform apparatus. Filiform apparatus. The cellular thickening seen in the synergids towards the micropylar end is known by the name filiform apparatus. Its function is to guide the pollen tube towards the egg during fertilization. This much about female gametogenesis and embryo sac development. Today we can stop here. Thank you.